Hello everyone, my name's Nathan Gray. I'm the host at Future of Microbiome, and I'm joined today by David Kyle again. This is a special on-demand question and answer session. Unfortunately, when David made his presentation, we didn't have any time to get through any of the questions. So David's very kindly agreed to step in and do an extra recording with us to go through some of those questions now. Thank you for joining me.
It's my pleasure to join you, Nathan. I'm glad to answer questions. It's always the fun part of the presentation.
Absolutely. So this probably is going to be about 10 or 15 minutes of just questions that we unfortunately didn't get time to go through in David's session at the Future of the Microbiome Congress. So we're going to start with a few questions relating to the study data that you presented.
Sure.
The first one, the first questions are talking about the fact that most of the studies that you mentioned are focus on increasing Bifidobacterium in infants, but there are also infants with other dominant species, for example, Bacteroides populations. So what is your opinion on the role or absent role of Bifidobacterium in these healthy infants?
Well, it seems like, as I had indicated in my presentation, in the absence of B. infantis to the breastfed baby and those HMOs, there are lots of opportunistic colonizers. We're concerned with the pathogens, the opportunistic pathogens, but the Bacteroides are generally considered commensals, but they're also opportunistic. We find them more abundant in adults than we do in infants. One of their roles is they are mucolytic, so they consume the glycans off of the mucin protein, and that collapses the mucus layer. And so that's not a very good thing to happen in the early infant. And so when we see, when we add B. infantis to those babies, the breastfed babies, and we get that elevated level of B. infantis, that lowers the pH, as I talked about, and it turns out that the Bacteroides are not really very happy at these lower pHs. And so it's either one or the other, it seems. It's either the Bifidobacteria or these opportunistic colonizers.
Okay. Thanks, David. We've had a follow-up question along similar lines, actually, here, which is ask you about how much of the inhibition of pathogens is actually because of B. infantis, and how much is potentially a function of some other things like reduced pH or other functions that are maybe alongside the treatments.
You know, we've thought about this a lot, and we've done a lot of studies to try and identify that answer to that question. And it keeps coming back that it's the reduction of the pH that seems to be the primary driver of the prevention, particularly the Enterobacteriaceae and those opportunistic pathogens. And we see that change in pH throughout the last 100 years. If you look at clinical trials where we've looked at baby stools and the pH of baby stools back in the 1920s, the pH was down at 4.8 to 5.2. Today it's 6 to 6.5. And so we think that correlation really shows that it's the lowering of the pH. But also remember, the microbiome is fueled by glycans coming down through the gut. And for the milk-fed baby, most of the good stuff is actually used by the baby. Thank goodness, that's what it's designed for. And it's only the HMOs that come through. So the consumption of that food source by B. infantis also prevents the growth of some of these other bacteria. So lowering pH and consuming that food source.
Slightly changing tack, but still relating to the findings of the studies a little bit. There's a question here asking about whether prebiotics and postbiotics during childhood could also have some of these effects linked to reducing asthma symptoms and things like that. Obviously, you're looking at B. infantis, a live strain, but could
Yes.
pre and post also work?
Well, quite frankly, I have not seen any evidence at all of any probiotic, quote, postbiotic treatment for asthma having much of an effect. I think most researchers are looking at trying to figure out how to prevent it from happening in the first place, which is what our focus is. And I have seen no evidence of any probiotics actually preventing asthma either. What we did show was that, or what others have shown, or Vuri in particular, was that a high level of gut inflammation in the first few months of life increase the risk or trajectory towards atopic dermatitis and asthma. And what we have found with the B. infantis is that one of its roles in terms of eliminating those enteric pathogens and acting as a colonization resistance, a natural colonization resistance, is to prevent that early inflammation. So I think there's a good reason to believe that if we can prevent that early inflammation in the gut with the B. infantis HMO combination, that could potentially reduce the risk of some of these autoimmune and allergenic conditions later in life.
Okay, thanks, David. We've got a couple of questions here along the same theme, I guess, of bacterial balance and diversity. So I'm gonna throw them together and see how they fall, if that's okay. So, is there a generally accepted optimal percentage for the dominance of B. infantis, and isn't sort of this overgrowth potentially of B. infantis actually reducing diversity of gut microbiome? You know, I think there's a few questions there along the same lines of whether diversity is actually being reduced, and what the percentage for optimal dominance is. You know, I think in one of the studies it showed it was about 80%.
That's a very good point, because that was an initial concern of ours as well too. Although if you ask the first question, which is what is a natural abundance of B. infantis in the breastfed baby, it is in the order of 70 to 80% dominant. And so when we ran our first clinical trial and reproduced that in a intervention model with babies, we found the same thing. But we now have metagenomics at our hand, and so we are able to look at the entire array of organisms that are there. And in our first publication of 2 or 3 years ago of that initial clinical trial, we showed that there really was no change in the overall composition of the gut microbiome. There is no change in the diversity. It's just that all of those other organisms were collapsed into a much smaller fraction, and it was dominated by the B. infantis. And so what we find is later, as that baby weans off of that breast milk and all of those HMOs, then the selective growth medium for the B. infantis is no longer there. And we see its levels dropping down, and other levels of the existing population that will form the more adult-like microbiome start coming up. And so it's just this very short period of time. It looks like the diversity is much lower because it's dominated by a single organism, but the diversity is still rich in those babies' intestine.
Sure. Now that makes absolute sense. One of the follow-ups here, and I guess you've already answered it, but I'm gonna just push on a little bit with it, is there a possibility that a lower dominance of, say, around 50% could still be beneficial, but actually give you a greater diversity and therefore be potentially even more beneficial for the infant?
Yeah, and let me answer that question in kind of a funny way, just to remind the audience that we're talking specifically about B. infantis here, and that there are many different species of bifidobacteria. And you will see in the quote healthy infant or toddler gut microbiome bifidobacteria levels in that region of 30 to 40% or something like that. But B. infantis may not be there. So it turns out that Zach Lewis about 4 years ago or so published a document looking at babies in Bangladesh, that there's this really bimodal distribution. B. infantis is either there at 70 to 80 to 90%, or it's not there at all.
Right.
And the bifidobacteria that replace it are other bifidobacteria that are not that good of consumers of HMO and are more plant glycan consumers that inhabit the gut later on.
Okay, great to clear that up.
That makes sense? Absolutely,
yeah. Really fascinating as well that it's either there or isn't. I mean, I'm gonna ask: is there anything you can share on sort of the potential mechanism of why that is? Why it won't settle a slightly lower percentage, or what the reasoning is behind that?
Right, and this may be speculation or not. I'm not sure, but remember, we only find B. infantis really at those numbers in the breastfed baby, and so they are getting and we have to remember that breast milk is it's a high fiber beverage. The fiber is there in those HMOs at 15 grams per liter. And if B. infantis is really the only consumer of it, then it becomes a selective growth medium and you get these very high numbers. So if the HMO is missing, you will not get the very high numbers. If the B. infantis is missing, you will not get the very high numbers because the other bacteria can't consume those milk oligosaccharides, and most of them go through and end up in the stool. So it's almost a black and white kind of situation. You have to have the combination of the two to build that high population. If you're missing the HMO, i.e. moving to an infant formula or moving on to weaning foods, then those levels begin to drop. So the key answer, I think, is that in those first 100 days of life, it's really important to have that B. infantis level or bifidobacteria level up at the 80–85% range.
Excellent. That's actually answered what was going to be my next question from
good.
from the next person, which was asking about asking if you could delve into a little bit how maternal milk composition regulates B. infantis colonization and whether this might have consequences for gut inflammation. I'll let you dig into that a little bit if you like, but I'm going to couple it to another question which is related, asking whether mother's diet can influence milk composition and therefore the baby's microbiome as well.
Good.
And whether you've looked at differences in mothers' diets, for example,
yes,
plant-based, non-plant-based, that sort of thing.
I can handle those two at the same time, because we have to remember that the bifidobacteria is there because of those HMOs. And so is there differences in HMO content within different breast milks? And so the answer, I think, the question is being asked is probably focusing on what we now know as secretor milk and non-secretor milk. This is milk that contains fucosylated glycans or so glucosylated oligosaccharides. A non-secretor will not have that FUT2 gene, so it does not contain fucosylated. The total level of the milk oligosaccharides are there almost the same. They're still both about 15 grams per liter in the mature milk, whether they're secretor or non-secretor. There has been some impact on the lack of those fucosylated HMOs on kind of a background, or what I'm going to call the control or the dysbiotic microbiome of the babies that don't have B. infantis. But once you provide the B. infantis, we established in that very first clinical trial that whether mom was a secretor or a non-secretor, the response was identical. That you could not detect a difference in the microbiome from secretors and non-secretors. Other components that will go into that breast milk from a diet to the mom has to be converted then into those oligosaccharides and change somewhat the oligosaccharides in the milk. And that really doesn't, I've not seen much information on whether a plant-based diet for the mother who's nursing that baby is going to have a difference on the impact on the B. infantis carriage.
Sure. Following on from that a little bit, I guess, a slightly different tack, and that is asking whether B. infantis, to your knowledge, or, again, it's probably slightly speculative, would be the only major colonizer in human breast milk, or maybe, I mean, part of the thing that you were talking about was the fact that this has been lost in a huge part of the population, and whether there are potentially others that have been lost in the past, potentially sort of pre-industrial revolution or even back to hunter-gatherer times. Is there potentially other major colonizers that have just been lost to time and could have been very beneficial? Sorry.
I'm sure there's been a lot that have been lost. I don't think we'd have to go back that far. I think the major losses started occurring when we brought antibiotics into the population about 60, 70 years ago now. I think that's where the most impact. And we know that if we go back into populations of the Yanomami tribes, for example, or these hunter-gatherer tribes, that their gut microbiome diversity is much more complex than ours living in America or in Britain or in some of these high-income nations of the world. So diversity definitely has been lost. There's no question about that. With respect to your first question of why B. infantis, here we can go into genomic analysis, and we can understand how these milk oligosaccharides are actually processed, consumed, and processed. And you have to have the genes in order to do it, right? And if you don't have the genes, you're not going to do it. And so there may be other organisms lost, but they probably didn't have the genes to consume these milk oligosaccharides. I do want to emphasize one point, though, because we, first of all, there's a whole range of genotypes within the bifidobacteria. We're just focusing on B. infantis. most people don't realize that there's a whole range within B. infantis. And a lot of commercial strains on the market today actually are missing some of those genes, probably not because of antibiotics at some time, but because of the long time cultivation of these organisms in a population or in a growth medium that wouldn't contain HMOs. And therefore they've lost the ability to transport these HMOs and utilize them. It's called the H5 gene cluster that's missing in many of these B. infantises. So not all bifidobacteria are alike, not all B. infantises are alike. Really
interesting. We've got a couple of slightly more speculative questions. Of course, most of what you've focused on is really focused in on quite young infants who are breastfeeding.
Yes.
Of course, and the clues in the name, that B. infantis is really, most predominant and important for people of that age group. Is there any data or potential mechanisms that you're aware of that would maybe show a benefit for B. infantis supplementation in older children or into adults even?
Well, I'm glad you made the point of mentioning the word B. infantis, because there has been a problem in the literature. So the first answer is yes. There's lots of literature on the B. infantis and impacts on children and adults. The problem is in the literature, the B. infantis that was reported was from a product called Align, made by Procter & Gamble, which was released all across the world, basically. It actually wasn't B. infantis. It's actually B. longum subspecies longum, not B. longum subspecies infantis. And so that is a longum longum. So all of that data that I've seen is with a longum longum, which has the genes for processing plant glycans. It's an adult, it's in you and me as adults, but not very competitive in an infant, which is getting only milk oligosaccharides. And so back to the point of the only way that you can build that large population of the real B. infantis in the gut is with supplementation of milk oligosaccharides. And right now, there's no source of that because we don't drink human milk. And that's really the only source of. But now with a lot of the production, large-scale production of milk oligosaccharides, human milk oligosaccharides, HMOs, ultimately to potentially go into formula, I presume, there's an opportunity to be able to bring this mechanism of action from the infant into children or into adults, maybe in a treatment modality. So we're working on that. I don't see anything published yet on that, and there may be some potential, but that is truly speculative, as you say.
And actually, those are the final two questions that I've got down here to ask you. Is firstly, and I'll couple them together again, and I'll leave you to take them in the order that you see fit. But the first one is asking about your thoughts for HMOs in general for adults as a potential novel microbiome modulator. A lot of people are sort of saying, obviously, that HMOs fit quite nicely into the prebiotic category, for example, and suggesting some benefits there. And also whether you see potential for co-feeding and co-supplementing B. infantis plus a HMO in adults for potentially for things like C. difficile or other conditions that are related to inflammation in the gut.
So I think you're right in terms of HMOs being maybe a whole new type, maybe even a quintessential prebiotic, as opposed to GOS and FOS, which are just short chain oligosaccharides that mostly consumed by everybody, because there is some selectivity, certainly for B. infantis, but other bifidobacteria who are not as good a consumer of all of the milk oligosaccharides, but may consume some of the smaller ones almost equally well to B. infantis, could increase in concentration in the gut. And so it's just, they sometimes refer to it as a bifidogenic, right? A bifidogenic factor. And so I think it is a very good bifidogenic factor. And I think there's some interesting applications that you can use there. You raise an interesting point with respect to C. diff, because you may or may not know that Clostridium difficile no longer sporulates when you get to a pH of less than 5.5 or so. And so if you could actually use this modality to lower the pH, you may be able to clear C. difficile. In fact, we had an interesting experiment. This is just as an aside, very high level, of some horses a foaling operation where the new foals were born, and there was an outbreak of C. difficile, and so these new baby horses were actually dying. They had hemorrhagic diarrhea, and many of them were succumbing to that diarrhea. And so the farm, because we're at UC Davis, called us up and said, is there anything you can do? I said, well, we can try B-infantis, because the mare's milk will also contain HMOs. And so when we gave that to the foals, it almost completely eliminated the outbreak of C. diff. So that's kind of an interesting finding in a different species that may suggest that there's some potential here for human therapeutics as well. Wow.
Unfortunately, that's really fascinating, and it opens up a whole. I mean, I could ask you questions on that for another half hour, but I'm aware that we're fast running out of time for this short Q&A session. So I'm in wrapping up. I'm just going to ask you to maybe highlight the most important one or two things that from your previous presentation and from our conversations today. If people only take away a couple of things from everything you've presented and we've discussed, then what are your real take-home messages for everyone watching?
Yes, thank you for that question, because I think that's very important, because we talked about a lot of very complicated issues and some really fascinating issues. I'd say there's three points basically, Nathan. First, B-infantis plus HMO provides a natural colonization resistance in the newborn infant gut. I think that was a major discovery and a really important one. Two, the fact that this natural protective mechanism for the infant has been lost in most of the high-income nations of the world just over the last 100 years through the unintended consequences of a lot of medical practices like the use of antibiotics and so on. So this colonization resistance is now lost. And as a result of this colonization resistance, we're seeing early chronic inflammation. And that may be the reason that we have this pandemic of autoimmune and allergenic diseases today. And I think that the last point is that not all bifidobacteria are the same. Not all B-infantises are the same. So we've identified a lot of strains, commercial strains of B-infantis that are missing that milk processing capability.
Perfect. Thank you so much again for your time, David. And thank you to everyone watching this on demand as well. A truly interesting initial presentation and a really fascinating Q&A discussion just now. Once again, thank you, David Kyle from Evolve. Thank you.
My pleasure, Nathan. Thank you very much.